Hey guys, hi, welcome. So today we are going to solve another question from the IBO 2013 paper. Now this question is very simple, looks very simple, but it's slightly tricky. Now what makes it tricky? Let's try to understand that. The following schematic shows a stage of cell division for an eukaryotic diploid cell. Now before I go ahead, I want you to just give a gist of the mitosis. So let's say there are 4 chromosomes. Okay, so 2n equals 4. So why I have drawn 2 chromosomes in the same color? The reason is that they are homologous. Homologous means one has come from mother and one has come from father. Okay, same way these are homologous pair, one from mother, one from father. Okay, in mitosis what does happen is all the chromosomes duplicate. So let's make their duplicates, the chromatids, right? Okay, so the chromatids are right here, and then all of them come at the metaphasic plate. Now in this particular demonstration I'm using colors to distinguish the chromosomes which must appear same. Okay, I'm trying to even make them look similar, but just in case. So these two are homologous. So when they are homologous, their morphology must be similar, right? They must look alike. Here I have used color to communicate that they are homologous, and the same is the case here. What will happen next? Very simple. The chromatids will be pulled apart. Okay, I'm trying to be less fanciful, so hence the line drawings. So this is the case of mitosis. I hope this is very clear. Now what would be the case of meiosis? Now let's try to navigate the same types of chromosomes for meiosis. So in meiosis 1, the homologous chromosomes come next to each other at the metaphase, right? Homologous chromosomes are here, and let's make this as well. So by the end of anaphase, or during the anaphase, the homologous chromosomes would have been pulled apart. So we have 2 cells now. And this one has this combination, and this one has that combination. Okay, now what we are going to do is we are going to take them through meiosis 2. So this was meiosis 1, meiosis 2. Now here what would happen? Once again these guys would be arranged at the metaphase. So let me arrange them at the metaphase. So this is for the cell number 1. Okay, so this is the cell number 1, this is the cell number 2. Okay, let me place them here. Now remember, very importantly, that these are non-homologous. They are non-homologous. So maybe the first one is chromosome number 1, the second is chromosome number 2. That means they will not be similar. Okay, so if you are given the picture from the metaphase, how do you recognize that they are from the mitosis metaphase, meiosis 1 metaphase, or meiosis 2 metaphase? If it's meiosis 1 metaphase, you will see the chromosomes, and anaphase, the chromosomes will separate. But in case of mitosis or meiosis 2 metaphase, what we are going to see is the chromosomes lined up, and at anaphase the chromatids would be pulled apart. Though if it is mitosis, there must be similarity in the half of the chromosomes at least. Why? Because all the homologous copies, they are at the mitosis. in case of meiotic 2 metaphase, there will not be similarity. Why? Because the homologous copies have already been separated. Okay, I hope you understand the difference now. Now let's get back to our question. Okay, the schematic may represent a stage of mitosis. Now, if you look closely here, the chromosomes, none of the chromosomes on one side even show similarity. Okay, so they are very different from each other. That means no, this is not mitosis, rather it represents the anaphase of meiosis 2. Now this was a trick, or this is the key observation that you need to practice in IBO questions. The cell would have failed to reach the stage if microtubular motor proteins were inhibited. Now, you know that the chromatids have been pulled apart because of the mitotic spindle, which is these proteins, okay, and then there are proteins here as well, and then this is where the crawling occurs, right? So this is where the microtubules walk on each other in the opposite direction in a way, and hence the spindle pulls apart the chromatids. If you do not have this motor proteins, if they are inhibited, then it will not occur. So this is true. Okay. Oh, sorry. Yeah. Green. This is true. This is true. And this is false. Okay. Transcription of histone genes peaks during this stage. Now, I'd like to remind you that right now we are in the cell division phase, right? And hence the chromosomes are in extremely condensed state. By condensed state means they are coiled and supercoiled. They are huddled with histones, which makes them not readable. Okay, they will not be exposed to enzymes right now. The transcription or of histones particularly, because we need histones for condensation, occurs during the S phase, right? When the cell needs them for further condensation. At this state, we do not have the readability anymore. Okay, so no, the answer is false. I hope this was pretty easy. Guys, do check out the book called NCERT Map. It's available on Amazon. Very soon will be on Flipkart as well. It has tons of material that can help you prepare and ace your exams really well. Thank you. Take care. Let's meet in the next one.